Thank you, Donna, for the invitation to join you this morning, and it's really my pleasure. It's been an outstanding week, and the meeting has been great, and this is just a great way to cap it off. I was asked to speak about genetics, but I took the liberty of expanding it a little bit more because I think most people who have a child with the disease, why they care about genetics is how can it help my child or the patient that you are advocating for. This is going to be a bit of an open discussion, and I kept hopefully will keep my talk relatively brief because I wanted to leave enough time for discussion because I cannot stay for the panel discussion. So I think it would be much more beneficial to you to have a discussion. So let me go through my slides and then we can have that discussion. So I'm going to make the assumption, and I know it's wrong, that you know nothing about genetics, and many of you know a lot about this, but just if it sounds patronizing, some of what I say, please forgive me, but I want to make sure we're all in the same field. So we're going to talk about some definitions, Biology 101, and then I want to talk about, there's a lot of buzz in the United States. There's a moonshot on personalized approaches to cancer therapy, and I want to talk about, what it is and what it's not. Have a frank discussion about the obstacles as well as the opportunities, and a vision for the future, and then time for questions. So, personalized medicine is not new, especially in the field of childhood cancer. All it means is an individualization of care, and as Donna just said, when a child's newly diagnosed, there generally is a roadmap that everyone follows around the world. But when relapse occurs or toxicity is encountered, we all personalize the approach. And it's been around for a long time, and I'll talk a little bit more about this in a second. Personalized genetic medicine is just the natural extension. So now that we have the ability to understand the genetic code in the individual and in the individual's tumor, it is how do we harness that information to further add precision to what we're doing. And that is not just about treatment, it's about diagnosis, and we'll talk about that a little bit, prognostication. Donna talked about NMIC. That was actually the very first, in any cancer anywhere, the very first genetic precision medicine, the very first genetic marker that told a physician in the family that there was something bad and you needed to adapt therapy. So it's been around for a long time. So there's personalized medicine and precision medicine, they're the same thing. So it gets a little confusing when we talk about genetics, genomics, and the way that I think about it is genetics are the study of the genes you're born with. What did you get from mom and dad? And genomics is the study of the cancer cell and what's gone wrong in the cancer cell. I think you all know that, and back what you learned in school, that when we're talking about genetics, we're talking about studying the double helix, the DNA, and here's highlighting a change in that DNA. And DNA turns into RNA, which turns into protein, and the protein is what we're targeting with drugs. What really has revolutionized our ability to gather this information is that 15 years ago, it took several $1 billion and about 3 years to sequence one genome. One human being's 3 billion base pairs, or what makes up a single cancer genome. And now we can do that for $1,000 in a day. And so there's been this incredible technological advancement which provides tremendous opportunity, but also challenges. And one of the things hopefully we'll get a chance to discuss is that I spend my life studying genetics and I don't understand the half of it. And for a parent to try to understand this complexity is really daunting. And maybe we could talk about how one can harness this information and understand it. So when things go wrong, there's a mutation. And a mutation just simply means any alteration that changes the spelling that encodes what your body's made up of. It gets complicated because we look different, our hair is different because our DNA is all different to start with. So our DNA already is different, so how do you distinguish a new difference, and what is the significance of that? And so this is a big part of the study of genetics is how do we vary, how do we differ from each other, and then if a mistake is made, is it really something that is promoting cancer? There are a lot of different types of mutations. They can be simple spelling mistakes, and I'll show you an example of that on the next slide. They can be where the DNA actually breaks and rejoins in a not normal way, which is called a translocation. Amplification is what happens with MIC-N and MIC, where there is supposed to be 2 copies of everything in our normal genetic code, and in the case of NMIC, for reasons that we're still trying to understand, in certain patients for incredibly complex reasons, it turns into 4 copies, then 8 copies, then 16 copies, and it can be hundreds of copies. And of all human cancers, where there are amplifications of many different genes in many different cancers, the NMIC gene is the most highly amplified gene of anything that's ever been discovered. So it's really quite incredible. There are nuances to this. The mutations can be germline in every cell in the body, inherited, or they can be restricted to the cancer cells, and that's the genomics thing that we were talking about. I'm not going to get into this because it gets very complicated, but there are nuances to this. The simple way to think of genetics is that the gene was bad in mom or dad, and you inherited that. But mutations can happen after the sperm and the egg join and be in only part of the cells in the body. That's called mosaicism. And in cancer cells, and this is something that we had an entire workshop on at the ANR, it is an incredibly complex ecosystem. We used to think that if there's a mutation, it's going to be in every cell in the cancer, but that's just simply not true. And one of the challenging things of deciphering the information of genetics is that it's often not binary. It's not just a simple yes-no answer. And so if there is a mutation, the question is it really an important mutation in promoting the cancer? Is it in every cell in that cancer? As you all know in this room, neuroblastoma has this incredibly aggressive behavior and spreads throughout the body, and we're only sampling one part of that usually. And the question is it in every part of the body? So this is just a picture of DNA and trying to show that these colored bars are the different bases, and that there's an original correct copy, and then there's a mutation. And this is something I stole from my partner Yael Mosé, who's done the work on the ALK gene, which many of you may have heard of, which is the most commonly mutated gene after MIC-N. And all of these different letterings down here are different mutations that have been discovered in children's tumors. So this was a study of over 1,000 children with neuroblastoma and looking for spelling mistakes in the ALK gene. And when you have any one of these, what Yael has gone on to show is any one of these mistakes that it promotes the cancer. And if it's in red, that's a kind of mutation that you can be born with. It's heritable. And if it's in black, we've only seen it in the tumor cells. So this is an important lesson for the rare cases where neuroblastoma is inherited and could it be passed on, and there's genetic testing that can be done to see if the child's at risk to pass this on, if they're a survivor, hopefully when they're a survivor, or if the siblings could be affected. That's rare, but something we can do these days. But as we'll get into, this can also inform treatment, and something that we're working on very hard. So the vision of personalized approaches to cancer care are grandiose in many ways, but I think very realistic. The idea is that we will be able to use modern technology to be able to more precisely define which patients are more likely to develop cancer, and then hopefully do something to prevent it. For those who are predisposed to develop cancer, and I think a theme that is emerging, as I'll show in the next couple of slides, is that the childhood cancers are much more genetic than adult cancers that are more environmental from smoking or sun exposure. So more children are predisposed to develop this. And if we can figure that out, then we need to do something about it. There's supposed to be, this was a mistake here, but we also can use this information to more precisely determine the prognosis, which means how to treat. You know, if the tumor is more likely to recur, then we should adapt our treatment. Where much of the effort has been in personalized cancer care has been in more rational and precise therapy. The promise of targeted therapy, that you have a mutant target that's driving the cancer, to have a drug to come in and kill that protein, and then the cancer relies on that and dissolves, is something that has been proven in some cancers. There is some proof of concept in neuroblastoma as well, but we have a long way to go. Personalized cancer care also is, once the disease is diagnosed, to be able to more precisely monitor the disease. So we've been doing this for a long time in leukemia, because leukemia cells are much easier to access than neuroblastoma cells. But many of you may have heard of so-called liquid biopsies, which are simply a blood test. And we know that neuroblastoma spreads through the bloodstream. And with the robustness of the technologies, we can now detect many of the mutations that are in tumor cells in the bloodstream. This still has a long way to go before it's common clinical practice. There are technological challenges, but there is the realistic possibility that in the not too distant future that blood tests can give us the same information that a tumor biopsy can give us. And then finally, there is tremendous amount of information that is emerging about, you know, what many of you may have witnessed. My child got very sick when they got cisplatin, but my friend's child got the same drug, did not get so sick. My child needs hearing aids, but the other child who got the same exact doses didn't. And so we are learning very quickly how these drugs that we use are broken down in the body, do what they're supposed to do in terms of killing cancer cells, but do what we don't want them to do to cause toxicities. And there is some promise that we'll be able to modify therapies based upon an individual's genetic profile. So as I said, and I'll go through this quickly, it's really not new. It is expanding rapidly, but it's really not new in terms of predisposition. We've known about certain genes that cause childhood cancers for many years, and there's been a lot of effort to prognosticate and to prevent, and I think that this has just all been exponentially increased as we've learned more. We've been, I already talked about the McN story, and that we've been using that as the very first example in any cancer to change what we do based upon a genetic test. Dinutuximab is a precision medicine. It targets a certain molecule that we can detect. Now, it's so common in neuroblastoma, we don't have a test and then assign the therapy, but as we get more and more experience with dinutuximab, that may be what we do in the future, is tailor the therapy based upon the actual presence or absence of this GD2 thing. We already talked about how in leukemia, which is easier to access, we've been able to monitor and change therapy based upon microscopic or submicroscopic amounts of disease in the body, and that is a promise with neuroblastoma, and that's something that is happening. And I already talked about the changing therapy based upon the individual's genetic profile. So this is a complicated figure from a recent paper authored by Rajan Modi, who is one of the participants, and this is, getting to the point of these are different childhood cancers with neuroblastoma here, and these are genes that we all have in our genetic code, and each of these different colors means that there was an abnormality detected when a child was diagnosed, the piece of the tumor, and then the blood was sent for one of these sequencing efforts where the DNA and the RNA was sequenced, and so a lot of alterations were discovered. The point I wanted to make on this slide and the next slide is that many of these are not in the tumor cell, but in every cell in the body, so they're inherited. And there were many other publications that came out at about the same time that all showed the same thing. So the important point of this, and these are all recent papers, right, came out in 2016, and these very important publications all had the same take-home message, that about 40 or 50% of the time, if you go through the effort of sequencing the tumor and the DNA, there is an abnormality detected that we think we know that we can do something about it, therapeutically or prognostically. The sad thing is that in 2016, that very few of the patients that participated in this were actually benefited from that information. And we'll leave time for discussion about this, because that is the real crux of the problem right now, is that we have learned a lot, we've discovered a lot, we think we know what we can do with some of this information, but getting access to the drug and access to the trials is the real bottleneck and the real problem in our pediatric cancer community. I'm going to click through this real quickly, because this was another paper that came out at about the same time that really emphasized the point that in the childhood cancers in particular, but in neuroblastoma as well, many of the mutations that we find can be inherited. Now, I don't want everyone running home to think that their child is genetically at risk, because it becomes very complicated, that it's not just one gene that you inherited. We're all familiar with a breast cancer example, that there could be a bad gene that you inherit and you can get breast cancer at an early age. What this is showing, what all these graphs and everything here is showing, is that many times it's more than one gene, it's more than one mutation, so it gets incredibly complicated. But the take-home message is that most childhood cancers arise from the genetics of what the child is born with, rather than an exposure that the mother had during pregnancy or the child had after birth. And so they're very different than the adult cancers. So as I just said, I do this for a living, and I'm awed by how naive I was a decade ago about how we would be able to figure this out. And every time we think that we've peeled a skin off of the onion, there are just so many other layers underneath there. So it is incredibly complicated. And as I hope we can get into in a discussion, is that this is why having a physician advocate that you can work with is so critically important. So we know that we've discovered over 1,000, and there's going to be many more spelling mistakes just in neuroblastoma that are involved with the disease. And how to figure out for an individual child which of these are important is something that we're working very, very hard on. But for an individual patient who doesn't have a family history, what we know is that it's probably dozens, if not thousands, of genetic mistakes that cooperate in this sort of perfect storm analogy. And so it's something that we're learning a lot about, but we're just, I think, still at the tip of the iceberg in our understanding. Once the cancer starts, we have now learned that these are ecosystems that evolve very dramatically. So we'll get into this in just a second. A biopsy done at diagnosis and a biopsy done at relapse, that can give you very, very different information, and I'll show you something about that. And that's very important to keep in mind. These are cancers. They're just like an infection. They evolve to survive, and that is the challenge that we're faced with. So as I just said, and I'll show in the next slide, we have to think carefully about where we get our information. So this is a complicated picture from a recent paper, and I just want to try to highlight, so this is a way that we sometimes draw genetic information where we take, you have 22 chromosomes and sex chromosomes. So this is your first chromosome, and then your second chromosome, and then your third chromosome. And then if there's spelling or lettering out here, this is a gene that resides at this place in the chromosome that was mutated. And so this was when a child who actually cared for when she was first diagnosed. And these were all the spelling mistakes, the mutations. Where there are lines here, that means that there were pieces of chromosomes that broke apart and stuck together. And this is a pretty typical neuroblastoma diagnosis. This child got chemotherapy, and then she suffered a relapse, and you can see we did a biopsy at the time of relapse, and you can see that there are many, many, many more spelling mistakes. There are many more translocations. There are many more changes. And what this bar graph is trying, and this is the same child right here, what the bar graph is trying to summarize for you is that if you just count the number of mutations in this child's cancer at diagnosis on the left or relapse on the right, there were about 40 of these spelling mistakes that were the same at diagnosis and relapse, but there were, another 50 or so at diagnosis that we didn't see at relapse, and then there were all these additional new mutations at relapse. And why this is important, I don't have that slide, is that many of these new mutations we've showed across this panel of patients activated proteins that we think in 2016 we know how to match with drugs to try to inhibit. So the lesson here is that if you rely on the diagnostic sample to inform relapse therapy, you may be very mistaken. You may be looking at mutations that are no longer there or missing mutations that have arisen. I think that many of you are your own children or those you advocate for are at major medical centers, and pediatric cancer is thankfully rare and needs to be cared for in major medical centers, or at least you need to have access for that for certain points in time, and we can discuss that. This is a child that early in my career, this is her relapse. This spot here on MIVG was a brand new spot. She had been in remission for some time. And early in my career, we would not even consider trying to get genetic information from this spot. It's a tiny little spot. We're not going to remove that section of her bone. It's in her thigh bone. But we have amazing colleagues in interventional radiology these days who can very safely put a needle into that area and return this, which is cancer cells. And so these are two dots that were put with a Sharpie on a slide. All this other stuff is debris. And we now have the way to not only diagnose this as cancer, but more importantly, to do genetics on this. And this is literally a fraction of a centimeter worth of tumor material. And there are many ways to get these sorts of information, which here don't mean a lot to you, but to me, this tells me that because this tumor has this genetic information, there's a drug therapy that we have that we would hope would improve this child's outcome. And this is a child who, his mother's been very active in many of these sort of forums and allowed me to use this, and she'll tell you that she had this procedure and was out dancing that night. So we can do these procedures safely. So in my opinion, we are now in equipoise. This is a term that Donna used, that we can safely access tissue at multiple points in a child's therapy to inform what we're doing. There are risks anytime you do a procedure, but I think that we're at a point now where we know that we can do it safely, and the information is of enough potential benefit that we should be doing more of these types of procedures. And as I mentioned, that there are many people, and there was an entire session at this meeting to hopefully get to the goal where we can do this in a blood test rather than a tumor biopsy. For the next 2 or 3 years, we're clearly going to be stuck with doing tumor biopsies. The good news, bad news about neuroblastoma is that it's often in places like the bone marrow where we can access it without too much trouble for the child. So in terms of obstacles and pitfalls about this, and hopefully we'll have time to discuss this, there is not a linear connection between the presence of a mutation and an effective therapy. This is a rapidly evolving field, and just because a particular mutation in a melanoma cell equates to benefit in a patient with melanoma doesn't mean the same mutation in a neuroblastoma cell is going to equate to benefit for a neuroblastoma patient. Because of the rarity of the condition, we're not learning as quickly as some of our colleagues with the more common lung melanomas, et cetera, but we are learning quickly. But we can't just assume that a drug that works in an adult malignancy is going to work in a childhood malignancy. Even more complex, and I showed that stick diagram from Yael Mosé with all the different ALK mutations. One of the most important lessons she's taught all of us is that it's not that the ALK is mutant, yes or no, but some of the mutations are poised to be inhibited by drugs that we have available today, where others are resistant. So it's not, and this is why having a physician who can help you interpret this information is so important, is that it's not just ALK yes or no, but what type of ALK mutation. And I think that this gets into something that this conference is so important, in the fact that this meeting is part of this conference is so critically essential, is that we need many more people across the globe who are trying to understand these connections between genetic change and effective therapy in order to make difference for children. So this is just another example that I wanted to show without getting into too much data about why knowing the type of mutations. So we've gotten very good at taking tumor cells out of a child with neuroblastoma, especially when the disease is relapsed, and being able to propagate those cells for testing in the laboratory. These are so-called PDX models, patient-derived xenograft models, and I'll focus on this one here because this was my patient Felix, whose parents wanted the PDX named after him. And this just shows how complex it is. This is how these grow in mice. So the very aggressive tumor, you implant the cells, and 2 weeks later all the mice have succumbed from the aggressiveness of this cancer. And we know that this disease has a mutation in ALK, and Felix did not benefit from the drug that we had available at the time, a drug called crizotinib, and the mice tell us exactly why. That the drug, this tumor was treated with this drug, it slowed it down, but ultimately progressed. So it's effective, but this is still progressive disease on a scan and progressive disease in the mouse, whereas this new drug, which is much more potent, effectively killed this cell, these cells, and cured these mice. And these are just different models to show how this works, and that even when the drug is effective for weeks, the currently available crizotinib, it grows back, whereas this drug is much more effective. So it is complicated, and you as parents and or advocates for children need to be able to partner with a physician who is up to date on these sort of information and can help you navigate. So there, as Donna said, there are over 100 phase 1 clinical trials potentially available. I would state the somewhat sounding pessimistic view that only a fraction of those have real data that support what's happening in those trials. And I think that you can get the information from responsible physicians about what is the evidence that supports why we're doing drug A and drug B, et cetera. And I think that there are charlatans out there. And you would think that this wouldn't affect pediatric cancer, but if something sounds too good to be true, it probably is. And so to help navigate this, everyone wants hope, everyone wants realistic hope, everyone wants an opportunity for cure. And my own practice has been, there's hope at every stage of therapy through the continuum. There's always hope. But that has to be put in the right perspective, and if someone tells you that they can take the tumor cell and promise you curative therapy, I think that's not an accurate scientific view of what really can be done. So it's, as Donna's talk, I think, very well put into perspective. To navigate this, you need partnerships and you need a community. So personalized precision medicine isn't just about DNA spelling mistakes. Immunotherapy is really personalized therapy, as I mentioned, with the dinutuximab. And there will be a day, I'm sure of this, where a child is not eligible for a clinical trial because they have neuroblastoma, but a child will be eligible for a clinical trial because they have a neuroblastoma whose tumor has a specific protein on the surface of the cell that we can target with an immunotherapy. And we already are developing trials that are designed that way, where it's not the diagnosis or the disease, but the biomarker or something that's present or not that makes you eligible. And then, as I mentioned, and I won't get into this in any more detail here, is that there's going to be a lot of future efforts in modifying how we give drugs based upon the patient's own genetic makeup to prevent hearing loss or other of the horrific side effects that we cause with our current therapy. So in the field of childhood cancer, there's an urgent need to prove that all this promise is real. And we are at that precipice now. This meeting was amazing to see how much advancement there's been in the 2 years since the last meeting. And there are many clinical trials that are opening, have opened or are ongoing right now that are really designed to prove the concept that we can get much more precise with our therapy. But the jury is still out. I'm a believer. I mean, this is what I do, but we still haven't proven that knowing an ALK mutation, for example, can make a difference for that patient. I believe that that's true, but it's going to take some time for us to prove that, but that's what we need to do now. And then I think that this comes back to, Donna talked about phase 3, phase 2, phase 1, but there's this continuum of research and learning from our patients in the clinic and harnessing that information for new discoveries and new therapies. And that entire, we have over 400 individuals dedicated to neuroblastoma research at this meeting. But there needs to be twice that number, in my opinion, at least, and we need to figure out a way to expand our efforts. So with that, I want to thank you for your attention, thank Donna for the invitation, and I'm happy to have a discussion, and it was great to see a hand already up, so thank you.
Thank you, John.